Thank you, Dan, for the introduction and for all of you for this invitation to speak with you today. Resilience is largely an ecological concept, and so when I use the term resilience with respect to evolutionary processes, it's using it somewhat loosely. But today, hopefully, what I hope to accomplish is to just review some of the perturbations that fish populations are experiencing, put that in the context of some theory that talks about evolutionary processes and the degree to which populations are resilient to these perturbations. I'll focus on some observations with respect to stocking impacts on brook trout in New York, which is what we're doing in the Hare lab, and then comment on what it means to manage for evolutionary resilience. So you may be thinking that, this is an evolutionary biologist, he's going to be talking about climate change, slow driver, long-term effects. But in fact, one of the main points I want to make today is that a lot of the perturbations, there's a lot of things affecting these populations and have been for decades, if not centuries. And so I'm not showing you projections of climate change in the future, but rather the average temperature changes over the last 150 years. And I just want to list here the many perturbations that are making large changes on what the populations are experiencing in their habitats, including fragmentation and non-indigenous species, hatchery-based stocking, and size-selective harvest. All these things together are contributing to a very different adaptive landscape than what the populations had evolved, had primarily evolved in. So, if we go to the cup and ball model, there's not a whole lot in evolutionary theory that suggests threshold effects in terms of perturbations that are pushing the ball towards greater extinction risk, and that unless we want to consider extinction the threshold, obviously that's a stable state. In conservation genetics and conservation biology, the focus has primarily been on a small population paradigm in which small population is accompanied by inbreeding and genetic drift, which leads to loss of genetic variability and inbreeding depression affecting vital rates, reproduction and survivorship, and this just spirals down. Okay, so this is a progressive and effect as you push whatever factors are pushing down towards small population size. But there's another perspective that I think is helpful today, in which, and I want to illustrate it with this really simple model, in which selection can act to help recover, a population from the spiral, or even more so when we're considering populations that are starting relatively large size, which population abundance is on the y-axis here, and it's a model in which there's an abrupt environmental change at time 0, and so we're looking at the trajectory of population size with several scenarios over time on the x-axis. After this abrupt change, what's causing the population size reductions here are selective deaths. Okay, so this is a very strong environmental change leading to a mismatch between the phenotypes adapted for other conditions and their current environment. So in some cases, the adaptive change is not rapid enough and the population goes extinct. In other cases, the selective deaths are causing selective directional selection towards better adapted phenotypes. Still, population size goes down into this risk zone. Okay, so the model does assume here a sort of a threshold effect, such that you're going into a zone where you have a much higher risk of extirpation or extinction. And the idea here is that with strong selection, it's not only driving you down into that risk zone, but it also is helping you escape. Okay. what's happening in this whole graph is that there's a gradient of selective versus the effectiveness of the efficacy of selection generating more adapted population, as opposed to the constraints on adaptation that you get in small populations when the random effects of genetic drift are the dominant evolutionary process. So I've been focusing on A, B, and C, which are sort of the trajectory of existing large populations that suffer an environmental change, but there's also examples, E and D, of introduced species that, again, are adapting to their new environment and either linking out or increasing. All right, so the take-home message that I want here is that there's no need to think about or wait for climate change to think about adaptive capacity, that there's plenty of perturbations that our populations are experiencing and have experienced. We need to be concerned about and try to understand the capacity with which they've been adapting to those. The other main point I want to make here is that management actions offer an opportunity to learn about these evolutionary responses. And so it's already been mentioned that in an adaptive management context, these perturbations, particularly the ones that we're imposing, like stocking, are an opportunity to learn about adaptive capacity in our populations. So what does it mean what's the spectrum of responses to environmental change? It includes these dispersal, phenotypic plasticity, as well as adaptation. And of course, extirpation is not a response, but rather a consequence if none of these are sufficient. Unfortunately, though, dispersal in a world in which we're increasingly fragmenting populations and putting up barriers has less potential to allow populations to respond to environmental change. Phenotypic plasticity is an adaptation itself, and one that allows for a great deal of flexibility in population responses. But it has both evolutionary constraints in terms of as an adaptation, and it has physiological limits, and there's cost, energetic cost to phenotypically, in a plastic way, responding to non-ideal environments. So with adaptation, it's adaptation via directional selection, as depicted here, is just one form of evolution that we want to consider today. And so I want to contrast it with the stochastic process of genetic drift, shown on the left, in which evolutionary change in allele frequencies, is due to basically the sampling error of moving genes from one generation to the next. And the smaller the population, the greater the sampling error, the greater the rate of random genetic change in that population. To the degree that a population is experiencing these random changes, that is a constraint on the ability of that population to respond to selection, okay? Because random changes can increase the frequency of deleterious alleles, and it can, by chance, eliminate alleles that would be adaptive under other conditions where there's not so much stochastic change. So, one of the reasons why I think it's important, it's great to be represented, to have evolution, have a chance to talk about evolutionary processes in a program like this, and important to consider evolution, is the fact that our recent experience, that is in the last decade or two, is very good documentation of rapid evolution in nature. And that's not just in fast generation time organisms, but with classic studies like in Darwin's finches. I thought I should give you a fishy example here, or at least a fisharian example, and I think a very good one in terms of rapid, good evidence for rapid adaptation is a case of grayling populations in Finland, introduced about 180 years ago, and then subsequently introduced to a couple of more lakes. And so the comparison here is 3 pairwise comparisons between 3 lakes with a maximum of 180 years since introduction into these lakes. And what we're looking at in the red bars is a genetic measure of the strength of genetic drift in these pairwise comparisons. And so that the x-axis here is either this statistic that refers to the amount of drift differentiation, FST, or for these other gray bars, we're looking at life history traits and the degree to which on the same scale they differ in these pairwise contrasts after accounting for and controlling for plasticity and maternal effects. So we're really looking at the additive genetic quantitative trait change. And so we see that those traits across these 3 lakes have changed dramatically and in very little time. And what's surprising about this example is that this kind of an introduction, where there's been a founder effect of a bottleneck size and subsequent bottlenecks into these other lakes, it's exactly the situation where you'd expect drift to have been a dominant force constraining evolution. So I do want to, I'm trying to show you that there's potential for very rapid adaptation in populations, but I don't want to give you the sense that there's never any constraints. Okay, there's also a lot of constraints, and for any one trait in any one population, it's not obvious the degree to which there's the capacity to evolve given a changed environment. So the specifics are very context dependent. My next point here is that evolution can be managed. Okay, so if it's happening on a time scale, an ecological time scale in which we're thinking about management, how do we affect it? Well, this is not new news. We can affect it by either reducing the amount of fragmentation among populations, allowing for corridors, or assist in the migration and gene flow by moving individuals around within metapopulations. Obviously, maintaining larger population sizes, and in this case, it's a genetically effective population size that matters for evolutionary capacity, is a means of minimizing the impacts of genetic drift and allowing maximizing adaptive capacity. So these are general recommendations that can either facilitate adaptive capacity, or in some cases, like in the hatchery, we actually want to constrain evolution. We don't want evolution happening. We bring in broodstock and amplify them for supplementation purposes. All right, so these general recommendations are a good foundation, but in any particular context, the adaptive response is going to depend upon the trait, the heritability of that trait, and the consistency of and patterns of selection. Okay, so so these are only going to get us so far, these general recommendations. All right, so I want to move to a specific case that I've been working on, and that is impacts of stocking of brook trout in New York. Starting generally, the expectations when using a non-local strain for stocking for fisheries enhancement is that if we're starting with a population that has natural genetic diversity that is a function of its evolutionary history and the demographic history of connectivity among the populations. So here in this cartoon, I'm drawing a divide or some kind of barrier that's led to genetic differentiation between these 2 regions, as well as differentiation maybe among these watersheds within region. And I'm depicting this in terms of neutral genetic diversity, so this would be indicating demographic history rather than adaptive differences. Okay? Traditionally, we've only had the ability with genetic markers to look at demographic history, and it's only recently with genomics that we've got the capability to also look at functional parts of the genome and ask genetically, even in non-model species in their environments, whether there's adaptive differences. All right, with stocking on top of these populations with a non-local source, consistently with the same source, our expectations are that we're going to homogenize the differences across these populations. But at the same time, any one population may end up with greater genetic diversity because now we're adding novel alleles from this non-local strain on top of the natural diversity. And notice that these differences suggest that there's different alleles in these different populations, right? So any one population has less genetic diversity than the whole, and now the genetic diversity has increased at the local level. So these expectations have been shown in several different cases. One really nice example is with lake trout, where comparisons of unstocked versus stocked populations have shown that a measure of genetic diversity, this is allelic richness, has increased at the local level, comparing unstocked to stocked. And again, looking at the statistic of population differentiation and comparing the unstocked populations with relatively high differentiation versus the stocked populations show much less genetic differentiation. Again, this is differentiation in terms of neutral markers, and it's interpreted in terms of connectivity or gene flow. So there's a greater amount of genetic homogeneity after stocking. So there's been some studies that have had the ability to ask with their data, what are the factors, the environmental factors, or the management factors that have been associated with a larger degree of introgression from non-local stocking sources into the stocked populations. By far, the stocking intensity has been the factor that has made the most difference where it's been looked at. But in addition, this case of brook trout in Quebec, it was seen that small water body and low maximum depth were important, as well as low dissolved oxygen and high temperature and pH. All these factors were associated with greater introgression. And it was speculated that this is not about how large the existing population was that was stocked on top of, but rather relates to the spatial diversity of spawning habitat that's available and the possibility that stocked populations that there's really little choice in terms of spawning habitat and therefore greater likelihood of rapid introgression under these conditions. All right, so in summary then, stocking with non-local strains causes dramatic changes in the neutral genetic structure of the populations. The functional significance of these changes, however, we just don't know. Okay, as I've said before, it depends upon the specifics of the adaptive traits that we're interested in and the degree of environmental change that the populations are confronted with. In principle, in theory, you might expect that the high genetic diversity that results from stocking with non-local strains may increase in adaptive response, but it depends upon the environment that they're adapting to and the degree to which the stocked populations are bringing in maladaptive alleles, perhaps from domestication selection in the hatchery. All right, so in New York, there's a long history of stocking for fishery enhancement, and with trout, that's including brook trout and non-native brown and rainbow. And I'm going to summarize a fairly complicated history in this way, fairly simply, with much of the early stocking with brook trout involving a domestic strain with a lot of different source populations contributing to it. But it was a strain maintained in the hatchery. Around the mid-'80s, there was much more emphasis, at least in the Adirondacks, in stocking lakes and ponds with F1 hybrids produced in the hatchery from crosses between the domestic strain and either a Quebec strain, Temiscaming strain, maintained in New York, or the domestic by a heritage strain, an unstocked heritage strain in New York called Windfall. And in addition, there's been some emphasis on using heritage strains, pure heritage strains, stocking those in reclaimed lakes to try to provide greater resilience through replication of these heritage strains. All right, so the diversity of brook trout in New York is phenomenal, and in terms of both phenotype and genetic diversity. And it's thought that some of that stems from colonization from three different Pleistocene refuges. So there's three different main drainages shown, and I'm showing that in color up here, that contributed to the population diversity and the genetic diversity in New York, either from the north in the St. Lawrence, or through the Hudson River and Delaware River, or the Allegheny. So that's so the natural diversity that we're working with. Our study, this is with Spencer Bruce, who's a PhD student at SUNY Albany and at the New York State Museum, my student Yuko Katsumi, and with Chris Van Maren at DC. We've been focusing on populations in this box, characterizing some of the unstocked and recognized heritage strains there, as well as looking at a series of stocked populations and genetically testing for introgression effects there. The study is motivated by these questions of trying to understand the ways in which heritage status matters. Okay, so is this simply a fact of trying to preserve evolutionary legacy, or does the genetic variation, the natural genetic variation that's still preserved in these heritage strains, is that like a stockpile of adaptive solutions, solutions that may differ depending upon which of these source populations we're looking at. Specifically, we want to look at the degree to which heritage strains are continue to be non-admixed, that is to show no impact from stocking or other contamination, and does stocking of previously natural populations lead to complete admixture? What is the degree of introgression? So we focused in terms of unstocked populations on these 5, and as well as these black dots represent the 26 stocked ponds where we sampled unclipped fish, meaning there were fish generated through natural reproduction in these ponds. So the first result I want to show you is a result on genetic diversity, and specifically heterozygosity, presented in terms of a standardized measure. So we're comparing heterozygosity across the study populations. And these study populations include, for each heritage strain, its natal pond, as well as one of the brood ponds where it's been introduced into for maintenance and easier accessibility. And so we've got Little Tupper, Windfall, and Horn Lake natal ponds, each with a brood pond. Onondaga, Wilmert, unstocked populations. The Temiskaming strain used for the F1 hatchery hybrids, as well as the domestic strain, and then 2 samples of these hatchery hybrids. There's well-documented difficulties in the Horn Lake natal pond with previous bottlenecks, so those are represented in this relatively low diversity. And it's also previously well-documented that the domestic strain, to be expected, has loss in genetic diversity, being a closed strain in the hatchery. But what's interesting here is that the F1 hatchery hybrids that are being stocked do show a trend towards higher genetic diversity that they're bringing into those populations. So the method of analysis that I'm using and I'm going to present results on here is a Bayesian clustering algorithm It's based upon a population genetics model, and it works with individual multilocus genotypes as the unit of analysis rather than with predefined population units. And so we can use this analysis to ask how many genetically differentiated populations are there among the samples. And here I'm asking that of the unstocked populations plus the brood ponds for these heritage strains. And the data gave the greatest support for 5 distinct populations here, suggesting that each of the brood ponds are not significantly differentiated from their natal heritage strain, which is good. That's the management goal, is we don't want evolutionary diversification between those. We want these brood ponds to be replicates of the heritage strains. And for the most part, relatively little signature of admixture. And so admixture I'm calling is these, each individual here is a line. So here you can see a different color. It's a individual multilocus genotype in which the algorithm allows for any individual to have contributions from any of these 5 populations. All right, so we see a little bit of admixture in the little tupper and in the brood pond for windfall. And I just went through all of those evens. So redoing the analysis now, without the brood ponds, but with the 5 unstocked populations, with Temiskaming domestic and their F1 hatchery hybrids, now we can ask what are the patterns in the stocked populations. And among those, we see, by and large, a pattern which is indicative of strong genetic contributions from the F1 hatchery hybrids that have been stocked for a couple of decades in these ponds. There's a few exceptions. There's some ponds that show influence from one or more of the heritage strains, and in Streeter, those stockings from Little Tupper are documented. For the others, some of the others, they aren't documented. And then the other interesting thing here is Greg population shows this pattern of a lack of admixture despite being repeatedly stocked with these F1 hybrids. So what could be contributing to that lack of admixture? Well, Spencer used multiple stepwise linear regression and environmental data on each of these stocked ponds from the Adirondack Lake survey, and to try to test for what environmental factors may have contributed to a lack of introgression in some ponds. Most of these ponds have had similar levels of high level of stocking, and so stocking intensity did not come up as a significant factor here. There's just not much variation in stocking intensity. But what did come up was watershed area to surface area. This is similar to what was seen in Quebec brook trout in terms of catchment area, and maybe an indication of population size, but more likely an indication of the spatial diversity of spawning habitats that are available, and that's affecting the degree to which stocked fish are interbreeding with the locals. All right, then, for managing to increase evolutionary resilience, or more specifically adaptive capacity, we want to go back to basics, and it's nothing new here that we want to maintain large effective population sizes. In a metapopulation, that means maintaining connectivity among population components. But in addition, we want to evaluate the fitness effects of perturbations when we can, and specifically with stocking and supplementation, we have the ability with every one of these events to increase our understanding of their effects and of adaptive capacity specifically. So we want to understand that in the context of the adaptive capacity and the genetic and evolutionary characteristics of the heritage populations, right? What does an unstocked population have in terms of environmental tolerances and adaptive capacity. Then does stocking affect these characteristics? We can measure the relative reproductive success as a function of introgression level, for example. And what is the geographic scale at which native diversity is important to conserve? Okay, so there's a lot of diversity, and if we're going to take a spatially explicit approach to managing that diversity, we need to know something more than simply how different are they. We need to understand, using reciprocal transplants and functional genomics, how different are they, and what does that mean functionally in terms of their relative adaptation. So I want to acknowledge the folks that are collaborating on the Brook Trout study with me, and my lab manager, Tim King, provided some of the Brook Trout data, and I've been collaborating with Cliff Craft on this. Thanks very much.
We don't have time for questions, but we are going to have a panel discussion this afternoon, so there'll be plenty of time to ask some questions, and we're going to take a quick